So, good evening to everyone. It's a pleasure being here. I'm talking here from Italy, and my place is at the University of Trento, which is small town, relatively small town between the Alps in Italy. So that's why this picture. So let me reintroduce you a topic of our research. And in particular, I would like to share with you some of our recent findings that we had on the topic of Kabuki Syndrome. So as you may know, our body is organized as tissues which are composed by multiple different kind of cell types. And each cell type in our body will encode the information for our being into the gene. Okay, so each cell of our body will have the same information, same DNA. But obviously, cells are different from one to each other, and they maintain these features even though when they need to divide, okay, like your blood system or your intestinal epithelia. So the question comes how these different phenotypes can be obtained and maintained through life, although the initial formation, so the genome, is equal in all the cells. And to understand this, we need to put into the frame the concept of epigenetics. So epigenetics, you can imagine as a layer of information which is beyond the genetic information, which is encoded by DNA. And the epigenetics is that sum of modifications and changes that are occurring at the chromatin level, but also partially also at the DNA level through DNA methylation, that allow the genome to be used to bring about the phenotype, meaning allowing some genes to be expressed or being repressed, depending on the cell type that we are looking at. So the important point here is that the epigenome regulates multiple aspects of our being, starting from the initial events of the development, okay, so organizing the embryos, giving rise to adult life, but also to other events such as stem cell differentiation, which normally would occur also in adult systems. And obviously, because the epigenetic regulation plays such a fundamental role, it is quite intuitive understanding that if the mechanism that regulate this layer of information is somehow altered, then the epigenetics alterations may lead to diseases, including Kabuki syndrome and other chromatinopathies. So how can we, let's say, think about it in this frame? Now, the epigenetic information is maintained through cell divisions. So you can imagine that you have multiple players that are playing around to build up this epigenetic state, working on this environment that is the chromatin. So they need to build up the chromatin and to maintain it and to transmit this information throughout the cell generations. And the important point here to remember that the chromatin is kind of a large biopolymer mass which is contained within the nucleus. And this is not a chaotic organization, but is rather a quite well and maintained organization. So just think that within each of our cells and each nucleus of our cells, the genomic information is 2 meters long, which is then organized in this structure called chromatin, which is wrapped up and organized within the nucleus. So there is a very high level organization there that has to be maintained. So the question is, what happens when this system does not work properly? The answer is that we ended up with a series of diseases, including chromatinopathies. Now, chromatinopathies can be seen as a group of rare genetic diseases, which normally are caused by the amplification of the genes codifying for chromatin regulators. These are generally a developmental disorder, meaning that the mutation may occur during development, and this acquisition de novo mutation gives rise to the dysfunction of the chromatin regulators. And commonly, these the diseases share specific clinical features. So reasoning that they may have common mechanism of actions by which their perturbation can lead to the outcome of these group of diseases. Now, here in this picture, you can see the results of this haploinsufficiency causing the alteration of multiple chromatin regulators, which are highlighted in this picture. And as you can see, they are, let's say, playing a crosstalk between each other. So they are communicating somehow, regulating the chromatin state. And so starting from this concept, we thought that maybe we could try to envision the role of these chromatin regulators using different biological systems. And so we started using the so-called mesenchymal stem cells, which are stem cells that are present during embryogenesis and also during the adult life, which are characterized by their capability to give rise to different connective tissues, considering also stroma, cartilages, muscles, adipose tissue, and bones, and so on. So they're very, very important, and are present in multiple tissues of our bodies, basically everywhere. And there are then a biological system in which we can try to understand the function of the alteration of these chromatin regulators. And in particular, I'm referring to KMT2B, which is causative of Kabuki syndrome. So let me drive you through what we have done so far. So we took these mesenchymal stem cells and we used let's say, engineering approach called genome editing approach, which allowed us to mimic the mutation state that you may retrieve in Kabuki patients. So basically, we reintroduced the mutation of KMT gene into this biological system. We did it on one single copy, so phenocopying what you would have at the genomic level in Kabuki patients. Okay, so when we obtain this biological model, we analyze them on different aspects at the level of looking at the phenotype, generally speaking, but also at the molecular level, looking at the transcriptome, and also looking at the chromatin level. And one thing that we, let's say, were focusing our attention was actually the nucleus itself. And before going into some details, I want to bring your attention to the fact that the nucleus is a compartment which is quite peculiar. It's highly dense. Within the nucleus, you have these functional compartments that are self-maintained, and they can bring about functions, meaning they can keep some genes in a repressive state and maintain them. So there is this chromatin compartment that themselves are sufficient to propagate a repressive environment that would not allow transcription to occur, or on the other side, to maintain an active state of the transcription. So our question was how the genome organization that I told you is about 2 meters is folded into this 3D chromatin structure is related to the genome function. And when talking about the genome function, you can think about the canonical, if you want, genetic function, so regulating gene expression, but also to other known canonical genome function, which we call known genetic function of genome. In particular, we are referring to the mechanical properties of the chromatin itself. Why this is relevant? Well, because the nucleus is the stiffest organelle within the cell, you can imagine that the nucleus itself has intrinsic mechanical properties, with the external part of the nucleus called nuclear lamina, which behave like an elastic module, whereas the interior part of the nucleus, which is composed mainly by chromatin and RNAs, have a viscoelastic response. Now, the nucleus is tightly anchored to the rest of the cells through this complex called the link, which physically link the nucleus to the cytoskeleton, which bring about all the mechanical forces that are governing the behavior of a cell. So we ask what could be the contribution of these chromatin compartments to define the nuclear mechanical properties and whether these are altered in the Kabuki syndrome. So we are not talking now at the genetic function of the genome and how this can be altered in the Kabuki condition, but rather the non-genomic function of the genome, meaning its mechanical properties. So what we discovered, I'm going to give you very, very few examples because I don't think that these are important, but I try to give you a message. What we discovered, well, you can see from these images below the cartoon that the nuclei in healthy cells or in our model of Kabuki are quite different. So in green, what you are seeing is the nuclear lamina, okay, labeled by lamina A, and in a kind of pinkish, you are going to see part of the heterochromatin. It's called polygon part of the chromatin. And as you can see, the morphology of this nuclei is highly altered. Okay, they are smaller, and this let's say different fusion of the nucleus is also associated with the increase of this heterochromatin state. This is not just true for our biological system, because we could retrieve the same feature also into fibroblasts derived, donated by patients. And what we find out more than that is that beside having this alteration in the nuclear organization, we have also an alteration on the nuclear mechanics. Without going to the details, the images that you're seeing here are retrieved by measuring the elastic properties of a nucleus. So the reddish means stiffer, the yellowish means softened. And as you can see, in a healthy condition, in the nucleus, you can clearly see the border of the nucleus as a nuclear lamina components, and these red spots at the center are the nucleus, which are components of the nucleus, which are quite stiff. Instead, in the haploinsufficient condition that we retrieved, this pattern is completely abolished. So you lose the stronger, stiffer part of the nuclear lamina, and you gain stiffness within the chromatin. And we think that this is quite relevant. And this is relevant because if the nuclear mechanical properties are altered, this impedes the cells properly respond to mechanical stimuli. So all the cells are under mechanical stimuli, like biochemical signaling, as have been shown before from my colleagues. And also mechanical stimuli are very important to guide the homeostasis of tissues, but also to guide the differentiation of the cells. So in our condition, what we saw is that the alteration of this equilibrium is quite dramatic. So you can see the nucleus in a very simplistic way, like candy within whatever your flavor you prefer, or if you look at the nucleus, you can imagine like a chocolate with a hardish external part and softened chocolate part inside. Okay, and you know that if you put this at hot temperature, that will melt, and they will not look like what you have bought at the beginning. Okay, now the nucleus is within directly linked to the cytoskeleton, and there are forces pulling the cytoskeleton here. So if the nucleus softens down, obviously the forces that are going to be brought about are going to have a different effect on the nucleus and vice versa. If you want even more simplify the message, you can think that in a healthy condition, the stiffness of the nucleus is kind of an equilibrium with the mechanical forces that can be generated by cytoplasm. So there is a continuous exchanges of information through this mechanical signaling. In the Kabuki syndrome, what we think that is happening, that we have a disequilibrium of these forces because of the softening of the nucleus, thereby now the cells are not anymore able to properly respond to these external mechanical stimuli. What that is going to cause? Well, this causes an incapability of cells to properly differentiate into, in this case, a chondrocyte and osteocytes. So what we did, we took our mesenchymal stem cells, we triggered a differentiation to our chondrocyte and osteocyte that would give rise to cartilages and bones. And what we observed that in the haploinsufficiency condition, because of the altered mechanical signaling, then the differentiation could not occur properly anymore. But then we focus our attention on a molecule, which is a protein, actually, which is called ATR, which works as a nuclear mechanosensor. What it means? It means that this protein gets activated and transmit a signal within the chromatin once there are conditions of increased nuclear mechanical response, stresses, sorry. So in this condition, ATR get activated. So we thought if we have conditions in which we have an alteration of the nuclear mechanical properties, maybe ATR is overly activated. And so we may rescue this phenotype by inhibiting its activity. And so there are compounds, drugs that have been already used for treating other diseases, which are specifically targeting ATR. So what we did, we used these ATR inhibitors and we assessed whether these could rescue the phenotype of the cells. And what we found out that actually the ATR inhibition can overcome the altered mechanical signaling, restoring the capability of the mesenchymal stem cells to properly differentiate into chondrocytes and osteocytes. This occurs also in vitro, because we measured that using our in vitro cell system, but that was not sufficient. And so we move our attention to a different model, which is a small fish called Medaka, which is suitable to investigate the occurrence of alteration of the growth of the animals and the alteration of the craniofacial aspects of these animals, because skeletogenesis and chondrogenesis occur very similar to what is happening in human beings. And so using this model system, what we did, we, let's say, phenocopy the Kabuki syndrome by targeting KMT2E also in this model. And you can do quite easily in these systems. You can do genetically, as we did, or with chemicals, and then you can proceed with drug screening. Now, in our case, we already have a target, so that was, a step that was not necessary. And then you can move, and similar to what has been shown to you with the C. elegans GFP animals, you can go and see what's happening during their development, because obviously this fish will develop quite fast. Within 3 days, you can follow the development and the organization of the cytoskeleton. And so we did that, and here the first pattern that we observe. So below you can see the, let's say, this is the head of the fish. You can see the black spot are the eyes, and you can see colored the cytoskeleton and the chondrocytes. And as you can see in the haplosufficient condition, the center picture, you can observe that the head is deformed quite easily, especially the front part. We measure all the size of these cartilages and the ossification stage. And then by simple treating with the ATR inhibitors for just 3 days, this was sufficient to restore while the animal was developing, so very early during development, the restoring of the normal facial features. And these animals could be maintained after treatment, healthy, and they could grow, and that was quite impressive result. Now, to go a little bit more into the mechanism, and then I'm going to finish. What we did, we let's say labeled our cells. So we use similar strategies that have been presented to you before with the fluorescent protein, but here we're labeling on one side cells that give rise to the bones, and the other one that give rise to the cartilages, okay, labeled in red and green. So as you can see in the wild type condition, you can appreciate during development that there are different cells that are participating in defining the future bones of the face of the fish. And in the haploinsufficient condition, you can easily see that there is a strong decrease of this signal, meaning that some potential differentiation of these cells is altered in the haploinsufficient condition. And by restoring the function of the mechanosignaling using the ATR inhibitor, we could fully rescue the phenotype. So getting back to a phenotype that was similar to the wild type condition, actually quite difficult to distinguish between the two of them. So to conclude, what I showed you that we move from an in vitro model to an in vivo system to study Kabuki syndrome using an engineering system to target the genome of healthy cells, so phenocopying at the genetic level what you would normally retrieve in Kabuki patients. And by analyzing the mechanical properties of chromatin, we could move from, instead of looking just at genetic function, looking at the mechanosignaling within this system. And so we really believe that mechanobiology plays a central role in explaining most of the Kabuki syndrome features. And you can think about that with respect to multiple aspects related to what you would normally observe in Kabuki individuals. So we really think that the gene codified by KM2PD, AMR4, play a fundamental role in defining the nuclear mechanical properties and in maintaining this, which is fundamental to allow cells to respond to the mechanical stimuli. And on the other way around, we can rescue these alterations by inhibiting the mechanosensor ATR. And so the next step would be to try to verify the preclinical potentials of our drugs using preclinical models for Kabuki syndrome, and then to eventually go to clinical trials if we succeed with this stage. So let me just conclude thanking all the members of my team and also the funding agency that support us in the last period to proceed with this funding. And thank you for your